Before you start, open up the notes link that was given to you. It'll look like this. You can do File, Make a Copy, name it something appropriate. I'd recommend sticking this in a folder somewhere in your Google Drive. Maybe start like a biology notes folder or something like that, just to help keep yourself organized. As we move forward, you're going to end up with a lot of different materials. It's probably going to be beneficial if you have those all in one place. Something along those lines will let you then have a good copy. Now that copy will open up. This will be one that you'll be able to edit then. When you get to that and you're going through, pay attention to these instructions here. You've made a copy. From here on out, this is going to be like notes that you'd get in class. You're going to see blanks. I've made them all bold and red. And actually it works kind of nice if, as you go through this on your own copy, you can double click on this, on the underscored blanks, and then you can type in your answer to that. So as we go through this, that'll help you to sort of keep your notes organized. You shouldn't have too many formatting issues and things like that. So to start, what we're going to be looking at is the different parts of the cell. Now prior to this, you've hopefully done the web quest that we did initially, which goes through the different parts of the cell. This will be repeating a lot of that information, but hopefully give you a pretty organized way to look at the different functions and parts of the cell and see kind of the bigger picture of how these things work together. I'm going to go through it kind of quickly, so if you need to pause, if you need to go back and do whatever it is that you need to do in order to have a good understanding of what each of these pieces do and how it works together to sort of enable the cell to be alive. Just a quick reminder of a few things that we covered previously. Robert Hooke is the dude who looked at cells for the first time. He looked actually at the plant cork, and what he noticed was a bunch of empty little rooms, which he called cells. So that's where the name came from. Here is the obligatory amazing picture of Robert Hooke. We also previously covered a little bit about cell theory. So the three parts of cell theory, just as a reminder, first off, all living things are made of one or more cells. Because of that, cells are the most basic unit of structure and function. If you look at any living thing and you want to understand what a particular piece or part of that living thing does, it comes down to the types of cells and what they're able to accomplish. And then the last one, which is pretty important, understanding that cells only come from existing cells. So we don't have cells that just, we don't have skin cells, for instance, that just pop into existence or things like that. Instead, they come from other previously existing cells. We grow new cells from existing cells. As we start this, and this will be the first few blanks to put into your notes, the cell is the smallest unit of an organism that's capable of independent functioning. So it's the smallest thing that you can look at that can exist on its own with relatively little interaction from other things. They contain genetic material, cytoplasm, and a cell membrane. I put this at the beginning just to be clear that all cells do these things. So all cells contain genetic material, usually DNA, sometimes RNA, cytoplasm, and a cell membrane. So when we look at that, that's true of every cell, whether it's eukaryotic, prokaryotic, plant, animal, bacteria, fungi, anything. Next, organelles. So organelles are the specific structures inside of the cell which have a very specific function and help that cell in a specific way. When we talk about organelles, in terms of eukaryotic organisms, they often have what we call membrane-bound organelles, and what that means is that each of those organelles will have its own membrane. So keep that in mind as we talk about different organelles. And then the last one, DNA, which stands for deoxyribonucleic acid, and that's the genetic information, which I think we know. The two types of cells, again, just review, there are prokaryotic cells, which do not have a nucleus. They only contain ribosomes when it comes to the list of organelles, and then our primary example here would be bacteria. Eukaryotic cells, they contain one nucleus, but they have many copies of each of the other organelles, and that's important to stress because some cells with specific jobs might have more of certain organelles than another cell. So for instance, you might expect like a muscle cell to have a ton of mitochondria because mitochondria provides lots of energy, and a muscle cell is going to be using a lot more energy than some random cell on your pinky toe. As we go through the different cells and organelles and their functions, you'll see on the first one for the cell membrane that it's already filled in bold there, sort of cell membrane. So on the left-hand side of your notes, make sure you write in the name of the organelle, and then there's some blanks related to what they do. Each one also comes with a picture, which should help you to sort of visually be able to identify what's going on inside of the cell. For each of these, though, I'm first going to propose sort of a problem that organelle is going to help to fix. So our first problem is, right off the bat, how do we have a cell what is a cell? How does a cell keep its insides separate from the outsides? Now, it seems like a silly question, but that's actually a big deal with how cells function. The whole purpose of a cell is that it can establish a difference on two sides of a membrane, and that allows it to do all sorts of chemical reactions, that allows it to do all sorts of actions that then facilitate the function of that cell. So for us, that first one is the cell membrane. The cell membrane is the thing that separates the inside from the outside of the cell. So here you have a very typical picture, clip art kind of picture of a cell. Here's the membrane. It regulates what comes in and out, so it controls what is able to come inside and outside of the cell. And it also just generally separates the cell from its surroundings. So stuff going on the outside is on the outside and generally doesn't come to the inside unless the cell wants that to happen or if something bad's going on. Important, this is made of fat. It's actually made out of phospholipids, type of lipid or fat. And recall that the whole thing with lipids is that they don't mix well with water. So in a very watery environment, the inside of the cell is very watery, the outside of the cell is very watery. This layer of lipid actually helps to establish that things can't freely move in and out unless they have the particular characteristic or particular part that the cell membrane is looking for. We'll go into cell membranes a lot more detail later on in another day. They also contain proteins in the cell membrane, and those are the things that are actually doing the carrying in and out from the cell. So our next problem, how can cells let things move around inside of it and not just all pile up in the bottom? Remember, going back to this picture real quick, the cell is a three-dimensional thing. We always see it as this little two-dimensional picture, or when you look in a microscope, it looks like it's this flat thing, but it is a three-dimensional thing. Gravity and all of that still applies. So it turns out inside of the cell we have what's called cytoplasm, and the cytoplasm actually refers to everything inside of the cell except for the nucleus. And it's a suspension. Basically, it's this jelly-like substance. It's mostly made of water, and it contains all of these different cell parts. And what that does is it helps provide a little bit of structure and support for these different organelles to sit within the cell. So how can the cell keep its genetic material protected, which is a big deal for cells? If you imagine the cells of your own body, you want to make sure that the DNA stays protected. When DNA gets damaged, things like cancer and other significant problems can occur, which can hinder the ability of your cells to do their jobs. Now, for us, we are a type of eukaryotic organism. Eukaryotic organisms have what's called the nucleus, and that's this other part, which is typically depicted as central in the cell, which makes sense because it's going to sort of have its hands in the control of everything that's going on around the cell. What it does is it controls the functions of the organelles, and it protects the DNA. So that is found only in eukaryotic organisms. A prokaryotic organism like bacteria is not going to have a nucleus, which is part of the reason why they are weaker cells in general. It's easier to kill a single bacteria cell if you spray some Lysol on the counter. It wipes out 99.9% of all of the prokaryotic organisms that might be existing there. If you spray Lysol on your hand, thankfully your hand doesn't just shrivel up and fall apart. And that's because we have a different type of membrane. We have our DNA protected within a nucleus. We're made of hardier cells than a prokaryotic organism. And one part of that is that we have a nucleus. What does the DNA actually do? What is it instructions actually for? The DNA is actually there to provide instruction to the ribosomes. The ribosomes are sort of the protein factory of the cell. They're super, super tiny. They're actually made up of little balls of RNA, which is another type of genetic material. And what they do is they receive a signal from the DNA that's essentially telling that ribosome that this is the protein that you're going to build. And that reaction that they're doing is dehydration synthesis. We saw this before. Remember, dehydration synthesis, removing water to create something new, something bigger. And so what the ribosome does is it takes individual amino acids, the monomer of proteins, and it turns them into different types of polypeptides or proteins and builds them up into that functional protein that's going to perform some task inside of your cell. So your DNA is sending that signal out to the ribosome, and there's a whole process to that. And that then is going to pick up that genetic material. It's going to read that genetic material and from that put together these different amino acids. Okay, so proteins build stuff in the cell. Where'd the ribosomes that make the proteins come from? Good question. And that's the nucleolus. The nucleolus is a region within the nucleus that's actually producing those ribosomes. Remember, I said that ribosomes are made out of RNA, a type of genetic material. So they're created inside of the nucleus and then able to be sent out. And that's where then they're going to do their job inside of the cytoplasm. So it's located inside of the nucleus. Next question, how do these cell parts get the things they need? How is stuff actually moved around? So we said that everything's sort of just suspended in this jelly-like cytoplasm, but you want to be able to move these proteins and things throughout the cell, and that's accomplished with the help of something called the endoplasmic reticulum. And as long as you write the word endoplasmic reticulum at least once in your life, I'm okay with you from now on referring to it as ER instead. Although it sounds pretty awesome to say endoplasmic reticulum in my opinion. So generally what this is the network of tubes that transport materials through the cell. There are two types, rough and smooth. The rough ER is the one that looks a little rougher. It's got these bumps all over it. The smooth ER, a little bit smoother. And what's going on there is that the rough ER is where ribosomes are actually attaching themselves and dropping off the proteins that they're producing. So the rough ER is called the rough ER because that's where the proteins are being dropped off. So if things need to be sent out of the cell, or if we need something special to happen to them, then what's going on? Because we've got this network of tubes, the endoplasmic reticulum, but sometimes a little bit more complicated things need to happen. And that's where the Golgi body, or the Golgi apparatus, comes into play. And what its job is to do is to package molecules that are going to be sent out of the cell, or things that need to do special jobs inside of the cell. It's actually made out of the same kind of membrane as the cell membrane, as the membrane around the nucleus, the membrane that separates the endoplasmic reticulum. It's all kind of made out of the same phospholipid stuff. And what the Golgi does is the endoplasmic reticulum can deliver these proteins or these products to the Golgi, and the Golgi will wrap them up in a membrane. And what this picture is kind of showing is like stuff kind of comes in, it blobs onto the Golgi and becomes part of the Golgi. And then also you get these little things called vesicles, which are just little phospholipid balls that contain maybe different types of proteins, maybe they contain enzymes, maybe they contain something that needs to be delivered outside of the cell. In the Golgi body, you can kind of think of it as like a post office of the cell. It's doing the organizing and making sure that things get sent off to the right place depending on what the cell is doing. So with all this building and moving, doesn't the cell just fill up with junk? Well, it would, for sure. There's lots of stuff constantly being produced inside of the cell. There's nutrients coming in. There's nutrients being broken down into their potentially waste products. There's lots of things going on. And that's why the cell has another organelle, which is called the lysosome. And the lysosome, real simply, it gets rid of waste in the cell. I like to think of it as like a little Pac-Man, just sort of going womp, 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 womp throughout the cell and eating up any sort of debris or waste products or really anything. Sometimes the lysosome's breaking down stuff that the cell might actually have wanted to use. The point is it has a very simple function. It just goes around and sort of cleans things up. It contains digestive enzymes that break down those unwanted materials, and it's made in the Golgi. So when we go back to the Golgi for a second, if we look at like these little vesicles popping off, if that's full of digestive enzymes, then we would actually call that a lysosome. So the lysosome is formed in the Golgi. It's just one of those products. Now the reaction it does is called hydrolysis. And we've seen hydrolysis before. As the name implies, it's using water to break something apart. So it's splitting water, putting an H back on one monomer, putting a OH on the other monomer, and you're breaking that back down from polymer to monomer. So as something that's doing breakdown inside of the cell, that's the process of hydrolysis. All right, moving forward. How can the cell possibly get enough energy to do all these things? All the things that we've talked about so far are very active processes that involve a lot of sort of moving pieces and things like that, and a lot of the different components of that require some energy in order to allow those proteins to complete their various jobs. Well, this is where we come to what hopefully everyone remembers from some life science in middle school, or I don't know, maybe they teach this in kindergarten these days, but the mitochondria is the powerhouse of the cell. Its job, real simple, is to convert food into energy. And that energy is a chemical called ATP, adenosine triphosphate, which is sort of the currency of energy that's usable within the cell. You notice it's not creating energy. It's converting the energy from our food into something that it can really create. So this should really say converts food into usable energy. The reaction is called respiration. And basically what happens is you have food, you have oxygen, and then that's going to break down into a form of usable energy, water, and CO2. So when I breathe out, that CO2 that I'm breathing out is because that process of respiration is happening inside of all the mitochondria, inside of all the cells in my body as they're using oxygen and food to produce the energy that the cell can actually use. All right, so what about cells that need to move on their own? So sometimes cells need to move on their own. Not all cells. The cells of my hand, for instance, don't need to be able to freely walk around my body. That'd be weird. But sometimes cells, especially in smaller organisms or single-celled organisms, need the ability to actually propel themselves. So there's two ways. Looking at this, cilia, which are tiny little hairs located on the cell that help it move slowly. And we see that in eukaryotic organisms that need to be able to sort of crawl along. Sometimes there's protists inside of pond water that need to be able to sort of crawl along their environment, and they can use cilia to do that. And then also in eukaryotic and prokaryotic organisms, they can have a flagella. Not all cells have either of these. Some cells don't have either one, but some cells have a flagella, and what that does is sort of like the picture implies, it's a long tail that helps to propel that organism rather quickly compared to something like cilia. And so it helps it to swim along to wherever it needs to go. How do cells store things? So cells aren't constantly needing a constant supply of all the materials in order to survive. That would be a pretty tough life. And so cells do have the ability to store various nutrients and water and other things, and that's happens inside of the vacuole. So the vacuole is a compartment. It stores waste, food, water, pretty much anything that's going on inside of the cell. It's found in all eukaryotic cells, but you'll notice in plant cells, in diagrams of them, you'll see a huge vacuole. It helps them to stay firm and to support their weight. So they have this rigid cell wall that we'll talk about, and the vacuole, as it fills up, basically like inflating a soccer ball, it pushes pressure on the outside, which helps the whole thing to stay rigid. If the plant dries out, if you forget to water your plant on your windowsill or something, it starts to wilt because these vacuoles shrink, which causes the cell wall to sort of squish in. Again, like a soccer ball or a basketball or something, if it goes flat, the sides start to push in because it doesn't have that internal pressure. So that's what the vacuole does for a plant. In general, though, for animal cells or other eukaryotic organisms, you'll see smaller vacuoles. It's all about storage of various things. So how does a 200-foot tree not squish under all of that weight? Well, sort of just spoiled that on the last one. So the cell wall is this thicker structure along the outside of plant cells. It contains holes, so it lets stuff pass through it. The cell wall is not really blocking much of anything, but the cell wall is really there for structural support. In plants, it's typically made out of cellulose. In bacteria can have cell walls as well. It's made out of a totally different material, peptidoglycan or other molecules that form the outside of the cell. Not super important, but the point is that a cell wall is really there for more structural support reasons rather than being there to prevent things from getting in and out of the cell. So a plant, for instance, just to be real clear, has a cell wall, but it also has a cell membrane underneath, the same phospholipid bilayer that we find in all cells. Finally, last thing, how do plants get the energy they need to survive? So plants don't have the ability to eat like you and me. Plants can't go out and get a sandwich. They can't uproot themselves and go run throughout the town and eat people. That would be terrifying. So what plants have instead, less exciting, is chloroplasts. And what chloroplasts do is they allow plants to make their own food. So the chloroplast doesn't really just give the plant the energy it needs to survive. It lets the plant capture energy from the sun in order to then make its own food, which is going to be glucose. Similar to how when we break down the food we eat, we typically break it down into glucose and other monomers. So this is why plants are green. They contain a pigment called chlorophyll, which is actually doing the capturing of the sunlight, another topic that we'll go into in more depth later on. And that reaction is called photosynthesis. So that process of photosynthesis is how plants take the energy of the sun, they take in CO2 from the air, and they take in water, typically from their roots, and they use those in order to produce glucose and oxygen. And if you're paying attention, if you go back to the mitochondria, you'll notice that the mitochondria is basically doing the exact opposite of this. It's taking a food source, glucose, for instance, oxygen, and it's producing a form of energy, CO2, and water. And plant cells have mitochondria just like you and me. A plant cell cannot get usable energy from the chloroplast. Instead, what it gets is the energy necessary to make its food. It then still needs the mitochondria in order to break that food down into the more readily usable ATP energy inside of the cell. All right, that was a lot going through all of those different organelles. Hopefully you've been able to fill in that note sheet. Hopefully that'll form a good foundation as we move forward and talk about different things that cells do. Make sure that you keep that someplace that you have access to it in Google Drive or print out a copy or whatever works for you, and then complete the exit slip that was posted. Thanks.